replication, mutagenesis, specialization, and cell division. Um, so yeah, as promised, course three. We begin with we begin with a little cartoon about genetic diversity, how it's adding a source of variation, like genetic variation, at the scale that enable population to adapt to environmental change. Like, yeah, that is obvious. But then when it comes to like species, all the way down to yeah, like the um, the fauna and flora, like the whole ecosystem. It you, you can almost link it to the extinction vortex. Like I'm not talking about inbreeding in humans. I'm talking about well, you could draw similarities and all that, but I'm not talking about like inbreeding in a narrow term. I'm talking about less forest, meaning less genetic variation, which means loss of genetic variability and blah blah blah, and small population all goes to zero. So yeah, like maintaining like a decent like niche for each species, and also yeah, finding methods to stabilize the whole mess of an ecosystem that we created, or that we like disturbed, is a go-to. So I can only it was you, B, right? You did have some worms that were able to downgrade plastic.
Yes, yes, I do.
Okay, well that's awesome because oh yeah, no, Arco Verde, yeah, we talked about even with small money, we can all kind of make a huge impact for real, like by buying a couple of those worms, making them grow, and
Yeah,
um,
I'm thinking
yeah,
about something with it, like videos, I don't know, to popularize these words in this objective, but and it's very small as is right now. So yeah,
right.
keep on.
Okay. Yeah. Okay. So a little last sentences before we go to the question. So the extinction vortex to the right is where a small population is prone to positive feedback loop of inbreeding and genetic drift that draws the population down to an extinction vortex towards smaller and smaller population size until no individual exists. One key factor driving the extinction vortex is the loss of genetic variation necessary to enable evolutionary response to environmental changes, such as the appearance of new strains of pathogen. Both inbreeding and genetic drift can cause a loss of genetic variation. And it will become clear what I really mean by inbreeding, but it's a keyword on that would be hybrid zones, like zones where usually you would have hybrid, like let's say butterfly with a yellow wing and purple wings, and you know in the hybrid zone they have like well, we'll get to that later. You have any question on that, or any comments, or you want to
Uh,
explain what you know about hybrid?
I
Yeah.
I don't know understand what is genetic drift in this context.
Okay. Genetic drift is you could almost say it's too much hybridization. It's like it's evolution has to follow like a fine balance between um, genetic variation and the how you call the healthier aspect of it, like not making children with African zebra or something as a human, like like just staying within your bipedal humanoid kind of thing, like that'd be an example of still being in the same breeding kind of I don't
Okay.
know. Like yeah, it's a fine balance. Like for having like a healthy genetic variability, you'd have to go in between those two. Like yeah, having not
Uh,
too much of a genetic divergence, and
Having someone too
yeah,
different to copulate, let's say,
right,
or
right.
to be even viable, let's say.
Well, I mean the you can have like I would imagine you could have something similar, like a more of a uh, uh, like a dissolution or like a like a vortex, but it's like more of a fountain, like something that just spreads across all spaces and just ends up being nothing. Like we would rather have like this helical structure or just like yeah, conserving all the good traits for say that we acquired and adding upon them with new ones from the ecosystem that's gonna be like more favorable for survival. Yeah. Any questions before we go to the good stuff? So from cell cycles or phases to the DNA sequence types that are categorized in the humans. So to the left, we see the like sort of a clock or like a pie chart where it basically paints a picture of cell division. What I mean with that is that in a dividing cell, you have the M phase, the mitotic phase that alternate with the interphase, and the first part of the interphase is called G1. So like it starts like it just divided. So you're the new cell that pretend you're just born as a cell. You're gonna go through G1, where you're gonna grow in size, making sure that you have like you're as big as the cool kids in the club. You have the DNA synthesis where your genetic material is going to be duplicated, and the G2 phase, which is going to be like a phase where you grow a little bit larger than the normal cell type, but it's just the final preparation for the final and repeating mitotic phase. Uh, yeah. So let's recap. We have cellular replication through division, where organisms they renew their tissue and organs, as we mentioned at like course one, part one, the first course. We kind of mentioned that. Uh, so we have one organism renewal of tissue and organs, and assuring genetic continuity through offspring. Are those the two only reasons? I'm asking just again. Don't feel forced to answer, but do do you know about other reason than those two?
Would you like to repeat the question one more time, please?
Oh yeah, cool. So cellular replication through division. I will list two of the 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 argument which makes it important and useful. Like, why is it useful for cell to divide at all? Like, if there was another way, well, would they choose that, or are there more than those two reasons that I will reiterate? So, first reason, organism renewal, tissue and organs. Like, basically, you have adult stem cells that's gonna divide and specify. Like, let's say skin cells, they when they reach the Hayflick limit of 30 divisions, they stop to be normal cell. Like they need to be like undergoing apoptosis or necrosis, depending on like the severity, but like usually apoptosis, and they self-destruct and give space for new cells that emerges from the adult stem cell. So that's the first part, organism renewal of the tissue and organs. And then you also want to assure that whenever like an offspring, like having like a genetic continuity through time, because we know that even diamonds aren't really forever, but if you can teach a man how to make diamonds, then you have something for an eternity. I don't know, but it's like, yeah. So assuring offspring and renewing tissue, are there more than those two reasons? Could there be more than those two reasons?
For
Like if an organism wants to expand, like I don't know, like a muscle, and it becomes bigger, and it has to produce more cells to expand in size, something like this.
Yeah, that is a very valid point. I'm actually gonna just say that, yeah, this is correct. It's narrowing the cell differentiation or specialization where embryonic or adult stem cell will differentiate and ensure that it not only has like the function recovered, but also like as a bodybuilder would do, like it would lay out a more robust, like let's say it would tell the specific surrounding cell that, okay, we need stronger pull force, we need like a thicker skin here, we need uh less or more sensitivity here, like like it the freaking body auto regulates like 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 no artificial intelligence or anything like we've ever seen before, right? Like yeah, and that's coming into play in the sequence that are like introns and regulatory sequences um, are those that's basically gonna code repetitive DNA include transposable element related to and related sequences. Those are also like uh, yeah, they're more fluffy than the introns, but still they matter, even though they they're openly saying like, we don't, you can just cut in our pieces, we're not going to affect much of the much of the gene. And well, basically, as you see that there is uh yeah, I think I cut it out, but it's like less than 1% of the genome is coding for stuff that we know about, like proteins and so on. Most of the gene we have is nonsense for now. Like we don't really know how much of it is epigenetics, like prone to acetylation, like prone to be silenced or opened, and so on. Yeah, man, I could talk all day about this.
I believe I had
Right.
problem on the
Did I just? Yeah, I think I might have been. This might have been. Oh, there you go. Yep.
So one quick question:
Yeah. Cool.
if 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 a cell is like replicating, like splitting, can it decide how fast it wants to duplicate, or like control if it needs to grow, and it needs more cells, so it can fasten up production somehow, or
Yeah.
how does that work?
Okay. Could you repeat the question? Like, I was doing two things at once. I'm sorry. Could you
Oh,
repeat the
no, no
question?
problem. So, can the cell like control how fast it wants to duplicate if it needs more cells, like increase the reproduction speed?
I don't. Well, I think it can. Yeah, I think there are. I worked with a colon cancer cell at some point, and we were given it the mTOR agonist and SIRT 1 to 5 agonist. Like it basically the mTOR was making them larger, but stopping at the probably like right between S and G2 phases. So you like, and they, because we were using like super proteins that are like native to their genomic or genetic ecosystem per se. We weren't really um affecting this cycle chemically, so like it kind of points towards, yeah, they can choose not to divide, but in in a dynamic um uh like, yeah, in an environment so dynamic as a like a animal body, or like a multicellular organism body, it really, you just want to follow what all the cells does, and you also get it. Like, it's very hierarchical. And yeah, I mean it probably would, you probably have like a way of increasing their growth and division potential by like up and down 10% speed, but they're also like, they're not going to divide if they don't have the the molecules for their succession. Like if they don't have accumulated enough, let's say, nucleotide building blocks, like the pyrimidine or purines, or like something that makes up the themselves, or a copy of themselves, they won't divide either. So it's also nutrient based, and then it, yeah, they also sort of die. So what was it, too much information, or did it satisfy your question, or?
Oh, thank you very much. It's perfect. Like, really interesting. But I think I don't. I hope it's not too much like off topic. Off topic.
Oh, no. It's actually perfect on topic. Like if no, the question of a cell being able to uh, uh, selectively divide faster or slower, that's a very good question, and I can. I just think it's very limited, and I'll get back on that. If you're there the next time I'm presenting, I'll try to have a more palpable response to that. But I appreciate the question. Sure, nice question.
Thank you very much.
Uh, they bud, so it's not really mitosis. It's a specialized form of mitosis. But they go through a reproductive phase, and the
Yeah.
reproductive phase is solely contingent upon oxygen being present in the environment. Once the oxygen runs out, they go into fermentation phase. They reproduce very slowly, and they get their oxygen by stripping it off of the sugar molecules and releasing carbon dioxide and producing alcohol, or yeah, and producing alcohol. So I mean the whole mitosis phase is governed by the availability of that oxygen, and when it's not there, they just kind of stop doing that and they go on eating.
Right. And that's yeah, that's very well pointed out, Sai, for sure. I was asking myself, like, are they not dying in a, let's say, minus Celsius environment. But no, like yeast, they're badass. They just continue on whatever, unless yeah, unless too drastic of environmental like chemical factors or something. They just continue on. That's also some cells are like that. Some others they like just a simple switch in magnetic field will probably kill them. I mean, nature is weird, but that's also kind of why we like her, or it, or him. Any question before we go to the mitotic phase of animal cell types? Because I realize I have a lot of things to say, and that's why I'm. uh, all right, so you have G2 of interphase. Uh, yeah, no need to go too much in detail of repeating what you can see, like nucleus, nuclear envelope, plasma membrane. Yeah. So I'll read up a bit on it. So yeah, so you have the nuclear envelope that starts bounded to the nucleus that will contain one or more nucleoli, and two centrosome that has formed by replication of a single centrosome will feature two centrioles. Chromosome duplicated during S phase cannot be seen individually because they are not yet they ha- they have not yet condensed. Oh yeah, okay, yeah. But apparently, so yeah, G2 of course, yeah, G2 they exited the S phase, so by the end of G2 they should have doubled their genetic material, and they popped up the centrosome ready to ready to start the mitosis. So prophase, the chromatin fiber become more tightly coiled, condensing into discrete chromosome observable by a light microscope. The nucleoli disappear, and each duplicated chromosome appear as two identical sister chromatids joined together at their centromere and all along their arm by cohesins, which is like the sister chromatid cohesion factor. It's um, microtubule meo something. The mitotic spindle, named for its shape, begins to form. It is composed of the centrosome and the microtubules that extend from them. The radial arrays of shorter microtubules that extend from the centrosome are called asters, stars. The centrosome move away from each other, apparently propelled by the lengthening of microtubules between them. That was many of these things I wanted to know Gabriel's input on, because like he mentioned something about like just those centrosomes, like those glowing magnets type of things. You apparently have like four semester worth of literature and courses to understand their function, like just those. So yeah, it's yeah. Prometaphase, the nuclear envelope fragments. That's when the microtubule extending from each centrosome can now like breaches and invaded the nuclear area. The chromosome have become even more condensed. Each of the two chromatids of each chromosome now has a kin kinetochore. Okay, I just learned one word. A kinetochore, a specialized protein structure located at the center. I will look that up. Some of the microtubule attached to the kinetochore, becoming the kinetochore microtubule. These jerk the chromosome back and forth. Non-kinetochore microtubule interact with those from the opposite pull of the spindle. Yep. Uh, any question before the? Well, we're halfway through animal cell types, and we have the plant ones. Are we good? Cool. Metaphase, longest stage of mitosis. This phase lasts about 20 minutes, where the centrosome are now at like at complete opposite poles of the cell. The chromosome convene on the metaphase plate, an imaginary plane that is equidistant between the spindle two poles. The chromosome centromere lie on the metaphase plate. Well, plate. Uh, Arguably magnetic monopoles, but shush, I didn't say that. Don't quote me on that. For each chromosome, the kinetochores of the sister chromatids, chromatids are attached to the kinetochore microtubule coming from opposite pole. Anaphase. It's the shortest stage of mitosis, often lasting only a few minutes. The anaphase begins when the cohesin proteins are cleaved. This allows the two sister chromatids of each pair to part suddenly. Each chromatid thus become a full-fledged chromosome. The two liberated daughter chromosomes then moving towards the opposite end of the cell as their kinetochore microtubule counterpart begins to shorten. When those because those tubules are attached to the centromere region, the chromosome move towards the centromere first, about 1 micrometer per minute. That is damn slow, but they don't need to go that far, you know. For them, let's say 2 micrometer. That's like a little jogging. So you have those cell elongating as the non-kinetochore microtubule link. Are we on the same page of what the kinetochore means? It's like they're the microtubules. They're the yellow rays. Uh, they're the kinetochores microtubule with some weird quantum effect or something. So yeah, by the end of the anaphase, the two ends of the cell have equivalent and complete collection of chromosomes, and that's where the telophase and cytokinesis begins. So in the telophase, the two daughter nuclei from the cell arises with along with their nuclear envelope, the and like the rest of the endomembrane system. The nucleoli reappear and the chromosome become less condensed. So that's the end of the telophase. That's where they're almost genetically um, indistinguishable from each other. Although if we're like that's where it has two roles of demonstrating that thing. It's also to okay. So it's either for like complete uh, analogous copy of one cell, like a daughter cell, or it could also be a specialization. Those two don't have to be the same exact cell. If it's a specialization, it one would still remain a stem cell if it's a stem cell, and the other one would become a mesoderm, ectoderm, endoderm, just more specialized tissue, and so on. Let's finish it with cytokinesis. The division of the cytoplasm is usually well underway by late telophase, so the two daughter cells appear shortly after the end of mitosis. In animal cells, cytokinesis involves the formation of a cleavage furrow which pinch the cell in two. Yeah, okay. Yeah, that basically, yeah. So we have like this, they they don't bud. They yeah, they how do like they cytoplasm their uh, membrane kind of to pop away from each other. That's that's basically it. Yeah. Any questions before we move to plant division?
So how long is this process like taking? What time are we talking? Like minutes, seconds?
Oh yeah. I'm glad you're asking because I had there you go. So the metatic phase approx 30 minutes, and the rest is also kind of where the hell did I put that? Yeah, let me make sure of that. But you can also, yeah, you can probably also just look it up on a quick search. But I would say the metallic phase is like around 30, whereas the G phase that's more or less. Uh, I think I read something about 24 hours for the G phase on a fast food intake, like on a normal intake, on average 24 hours for the G phase. Then the S phase is like approx half a day, like 11 hours, 10 hours, and G2 something around. I don't remember the G2. I think the G2 can be instantaneous if they need to, but that's actually where they choose kind of with the
I see.
as I mentioned, yeah, the M tour, like the
Oh, okay, thanks.
mammalian, yeah, yeah.
Is it going to
Okay.
matter specifically what organism we're talking about?
I was thinking, yeah, mammalian cell, human cell kind of thing,
Well,
because that's what I work with.
some bacteria can reproduce rather quickly, and
Oh yeah,
human cells I think are much slower. But there's a lot more going on in our cells. So.
oh yeah, oh yeah. Like the rate of division will 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 vary like extraordinarily from species to species. If that's something I'd agree with. So mitosis in a plant cell. So we have light micrographs from uh, the mitosis happening in an onion root. So in this onion root, we have the prophase. Uh, yeah, like the chromatin condensing again, beginning disappear. Although not yet visible, the metallic spindle is starting to form. The prometaphase, discrete chromosome, now visible. Each consists of uh, two identical sister chromatids, and they kind of have to be identical at this point. Uh, the the nuclear envelope is about to fragment. Um, the metaphase, that's where it actually like bursted. The spindle is complete, and the chromosome attached to microtubule at their kinetochores. Kinetochores. Yeah, I swear I must have learned this word at some point, but I completely forgot about it. Or so yeah, like at their kinetochore, they're all like at the metaphase plate or orientation. NFA's chromatids of each chromosome have separated, and daughter chromosome are moving to the ends of the cell as their kinetochore microtubule shorten. And the telophase, daughter nuclei are forming. Meanwhile, cytokinesis has started. The cell plate will divide the cytoplasm into and is growing towards the perimeter of the parent cell. Yeah, quite spectacular. Any question on that? Good, good. Okay, the coefficient of relatedness, the relationship coefficient between siblings, as drawn here as with parent A sharing one genotype dash phenotype. If you know it's, for example, uh, diabetes, uh, then let's pretend the red marker is diabetes, and you'd have like a 50-50% probability of it being transmitted to the next sibling. That's the basic of Mendeley. Uh, so a sibling has inherited the allele from parent A. There is a probability of 50% that sibling 2 will also inherit this allele from parent A. Any allele present on one chromosome of either parent will behave similarly, so the coefficient of relatedness between those two siblings is thus one and a half. Yeah, and that's then you can continue the graph. Let's say that uh you can it's like it's all dilution or confirmation that a gene is dominant or recessive. If the gene disappear of like not disappear, but if the gene is not expressed anymore, all of a sudden it's but has been and will probably be because history tells that that means it's a recessive gene. If it a gene like if it's a phenotype that is considerably there, like and no matter if it's no matter how many children, it's always going to be there. Then it's a dominant gene. Uh, yeah. So the chromosome, you have a three-step process on like how to like how to visualize its duplication and distribution during cell division. One eukaryotic cell has multiple chromosomes, one of which is represented here before duplication. Each chromosome has a single DNA molecule. Once replicated, a chromosome consists of two sister chromatids connected along their entire length by sister chromatid cohesion. Each chromatid contains a copy of the DNA molecule. Um, and then three mechanical processes separate the sister chromatid into two chromosomes and then distribute them into daughter cells. Like it's big. It may come as completely obvious here, but it's also kind of to get that clutched in when we talk about plasmid. I promise there is a plasmid shown at the end of this, and that's gonna be interesting. Yeah. Okay. Any question before we move to? No. Are you awake? Yeah. You there?
Yeah, of course. Still
Okay.
listening.
Good. Good. I was just being afraid of me talking to myself. I have
Yeah, yeah,
I've
no
had
problem.
this happening. So okay. Um. Good. So determination of the determination is basically a term for a precursor cell or an embryonic cell to specialize. It And in this case, for example, you have like an embryonic precursor cell signal um, that's gonna get a signal from other cell to lead an activation of a master regulatory gene called MyoD, and the cell makes MyoD protein, and the cell to make MyoD protein specific transcription factor that acts as an activator. So this cell is gonna basically, yeah, say, yo, I just came here. How do I graduate? And they're telling it like, yo, you basically grab that part of the DNA, our chromosome or chromatin, and you just gotta activate this, which is done with the help of MyoD protein transcription factor. Um, so yeah, at the determination, it just turns it on. It's gonna make some messenger RNA be that's gonna be yeah. Oh, it's many information at once, and I'm trying to make it as simple to understand as possible. So I should also just uh, okay. Did, did that make sense so far? The determination.
Like sort of is, I guess.
Okay. So yeah, like when it gets told when the baby precursor cell of a muscle gets told, yeah, you're basically gonna become like 100% neoblast muscle cell. So yeah, have a go at activating MUD, and we'll see you at the differentiation phase. And yeah, that's basically where where it like it gets committed to become a skeletal muscle cell. Number two, at differentiation, the MUD protein stimulates the MUD gene further and activates gene encoding other. Oh, okay, yeah. It activates some other genes encoding other muscle-specific transcription factors, which in turn activate genes for muscle proteins. And MyoD also turns on gene that blocks the cell cycle, thus stopping the cell division. Well, yeah, I would assume it stops its own. So, like, yeah, it's like, okay, we determined that we need to be this. We don't need to differentiate more. We can basically, yeah, we can start paying off our mortgage and stuff. So the non-dividing myoblasts fuse to become mature multi-nucleate muscle cells, also called muscle fibers. That's it. The guy is set for life. Uh, yeah. Any question on that?
Um,
Hmm? Oh, thank you. I'm trying to make it understandable. it's it's a challenge, but I like it. Okay, so here we are at the hybridization zone, and that's like that is quite something. um, so yeah, well, going back at biodiversity and how um like too much inbreeding would cause um ah genetic too much genetic what genetic fucking pardon my obscenities. Yeah, drift. There you go. That was it. That was genetic drift. So that is like a way of nature to automatically counter too much genetic drift, but also too much inbreeding. That's having this um, population that automatically will start to diverge until some sort of a specialization, like the gene flow. I suppose they mean reproduction. They haven't like said that really, but like yeah, the gene flow, it has to do with passing your gene on to the other ones and maintaining this zone of possibilities of opportunities. Uh, and yeah, I mean, like nothing that I would read up here would sound like groundbreaking or mind blowing. Like it's a thing that is proven over and over again that one should not uh, be inbred and stuff. Like yeah, it's same old story, but it's like with convenient pictures and it's on the slide in the link. You can grab the slides. Just just don't forget to credit Wikipedia, Campbell and Reese, and Caltech if you would use them in a non-lucrative manner. Else, I'm not responsible for you using this for making money because, like you said, shit. Okay. Any question before we go to the other bit?
I think we can go on.
Sorry, I was speaking on mute. Sorry.
Oh yeah, I love that.
Can you go back on the slide?
Yeah,
How does it fight genetic drift? I did not understand the mechanism of
actually, this is like to add a controlled genetic drift. It's basically to fight um, in inbredness. It's like the creation of a hybrid zone is to fight the decline in gene um like gene individuality, but like also like having gene variations. Yeah, that's a better word, like gene variation uh
so
and population. Yeah.
okay, yeah, I got it. They one from what I understand.
Oh, you're right though. You're right that there is definitely like yeah, there is a reinforcement part where like there are three or several possible outcomes for the hybrid from there. Like they have made a hybrid zone, but they can form a gene barrier, stuff that makes them unable to reproduce. Like if you cross a zebra with a donkey, no, like a horse with a, you can actually have a horse fertilizing a donkey, and that will make a mule, but the mule is sterile.
Uh,
So yeah, it has like basically a barrier
so
that prevents the
you is the hybrid zone or is
the mule is in the hybrid zone and it cannot like cross any of the barriers because it's made sterile for now until some other type of
fuck it up?
horse or some other type of of donkey will create a new type of mule per se. That's how nature works.
The new example was also
Yeah,
we could have a meal that let's say, wow, I will be reproductive. Thank you.
yeah, that could happen. That 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 happens. But yeah, barrier to gene flow that can be like, for example, uh, they don't like as a as a collective consciousness, they don't consider the other members of the group attractive anymore. All of a sudden, they do consider them attractive. That'd be like lower the barrier, and then you have like more physical or somatical factors such as sterility and so on. Yeah,
Okay,
so you have stability, which is continued production of hybrid individuals. That's kind of known as stable because you have like a stable flow of genes. You have fusion, which is that's yeah that yeah it just obvious stuff. Well, I don't know. For me, it kind of is. Do you want something explained more in detail or
No, I'm fine. I think I got it.
cool, cool,
Thank you.
perfect, awesome. So now comes with uh the other part. So you heard of the Yamanaka cocktail, the stem cell time clock, and like how like the research done that you can basically tell one of your adult stem cell to go back in the embryonic phase, which is nice and all, but as Gabriel said, it's not it's not enough to have like an on and off button, because the on and off button cannot be adjusted specifically to reach the center, per se, of this, or like the opposite center of this. Like you want like a, a more stable and regulated cell division, which will lead to less chances of contracting cancer, uh, because increased metabolism, aka division, and so on, will always lead to an increase of potential, um, for melanoma and so on. So that's that's an oncogene, and we don't want that. We don't want to overstimulate um, the cell cycle. We don't want to overexpress protein, but we don't want to neglect them, or like deny them access to some nutrient. This is like unless you're yeast and you don't really care about anything. Like JPL, a very good buddy of mine, is gonna present the actual plasmid club, or he's gonna lead, and hopefully will. Well, JPL. He told me one time, and they're always gonna stick with me. Like the yeast cells are frustrated teenagers. Like the way they conserve genes is like if it's useful, they may take it up. If it's not, they definitely throw it out. Like they, it's like I don't need that. Uh, yeah, but okay. So the cell cycle stimulating pathway, you have this pathway triggered by a growth factor that binds to its receptor in the plasma membrane. Of course, the signal is related to the G protein called ROS. Like all G protein, ROS is active when GTP is bound to it. Um, it's guanine guanine triphosphate. It's an analogous ATP, but it's not quite as charged. So RES passes the signal to four a series of protein kinase. The last kinase activates a transcription activator that turns on one or more genes for protein that stimulate the cell cycle. If a mutation makes RES or any other pathway component abnormally active, excessive cell division may end up in melanoma or cancer. Uh, so that's how RAS can affect the protein kinase in the phosphorylation cascade, and make it snowball so hard that it just crashes the whole thing. The cell cycle inhibiting pathway is the other way around, another extreme, the opposite. DNA damage is an intracellular signal that is passed via protein kinases. When they sense that some DNA has been damaged, the the kinases, they're gonna activate themselves,
Mm-hmm.
and in turn it would lead to an activation of p53,
Oh, right.
yeah, right, um, which promotes transcription of
Yeah,
the
I'm
gene.
gonna just
Oh,
Isolate.
Gabriel,
yeah.
Yeah.
Hey.
Are you with us or are you in the phone?
Oh, Gabriel,
So,
gonna say something.
Hey, bro, if you are with us, I don't know, shake a water or something. We grab a peso here. Are you with us? Charlie?
Maybe something with the
just go ahead and throw it at him.
Yeah, he's definitely not with us. He might be on
Mhm.
the
good stuff. You want me to continue? You want me to?
Do we just mute him?
I probably will.
No.
Okay.
Let's wait.
Wait.
Because I don't need to mute him.
No. Now Gabriel, he's gonna say something when he's got something to say. That's how Gabriel works.
Yeah, but he's not speaking with us right now. Are you
No,
back?
it's.
I believe he.
I think
Oh,
it's the headset. He's got some headset issues. Sometimes it. Yeah.
I
So
believe.
yeah.
I he. Okay, yeah, he left. Now,
Okay. Okay. Yeah. So P53 promotes transcription of the gene inhibiting yeah, okay, it inhibits the cell cycle, which is quite good. In that retrospect, it can just go on the extreme, and because like all it does is like, oh shit, we gotta stop the division, because if it divides, it may be a cancer dividing, so we just stop the division. And I hope, fuck, Gabriel, you would have supplemented with good knowledge. Well, yeah, that's all I know for now. That's all I've been researching. It's that, you know, P53 is gonna inhibit the cell cycle, resulting in suppression of cell division, and ensuring that the damaged DNA is not replicated. Because mutation causes deficiency in many pathway components that contribute to the development of uh, cancer. Again, any question on that?
I don't
Good,
think so. No.
good. So we have the a nice little handy term uh, with people such as me learn it: oncogene and proto-oncogene. Any idea on what it means? Sorry, what?
it's very low.
Yeah, I have
Sorry.
a hard time. There you go. Try again.
Uncle Gene and Aunt Norma.
And proto-oncogene, oncogene and proto-oncogene. Oh, you made it like I don't, I don't know. Well, basically, oncogene are those that will, yeah, that are carcinogenic. An oncogene is a carcinogenic, ge- um, or a gene with a carcinogenic supposed effect. Namely, yeah, it will induce too much stress, or it would like yeah, degrade stuff left and right, or it would, like just signal excessive growth or so on. Like it just likes to not do normal things. That's an oncogene. Like, and yeah, it and the proto oncogene is its direct opposite. It's like It's a gene that is supposed to be there to protect against oncogene. But yeah, some genetic changes such as sunlight or you name it, like car exhaust, cigarette smoke, you, factors can turn those proto-oncogene into oncogene, and yeah, that's something to like look out for in any environment.
Can you repeat how it works?
Okay, so a proto-oncogene is a gene sequence um, specially tailored for preventing oncogenes. Like a proto-oncogene is um, to like it's they're there to prevent mutation and so on.
Okay.
Oh wow, what is okay? Just like a Discord is having a class right now, but it's like yeah, I'm sharing the internet to my computer with my phone. I'm talking through my phone. Gosh, I'm
Yeah,
yo, but it's Friday the 13th, so we gotta do the do.
what gotta do?
Come on, are you freaking? Yep. Oh, this is marvelous. Guess what, boys and girls?
Or the universe decided that you're ready
Oh,
to
there
end.
you go. Ah, we got it. We got it. We got contact. We got. I believe. Come on. Yes. There you go. I only have like 2 or 3 slides left. I think it's. Yeah. Can you all see this?
Yes.
Good. Good. Okay, 2 slides left. So here's to the left we have immunoglobulin, like an antibody that this one loves to shuffle its own geno like genomic arrangement up. Like of course it's a protein, but it's full of like it's coated with specific gaps, full of introns that's gonna yeah like it's a game of hide and seek right out there, or like a game of hide and seek and tag and all sorts of foul plays in the world of virus detection versus um, immuno uh, camouflage, or like like it's basically can you find me, or can I find you finding me, or some
Cat and
wicked.
mouse. Cat and
Yeah,
mice.
it's like it's some crazy mind games, and for crazy mind games, you need a quite a nice meta, if you understand the term. Like so, this is like the perfect way of ensuring uh decent meta game is to shuffle its receptor uh structure so many times that it's gonna still act the same because it's controlled shuffling, but it's still gonna be different for other that didn't catch the memo, which is the viruses. And yeah, um, a little word on it. It's the joining of randomly selected B and J gene segment in this example, result in a functional gene that encodes the light chain polypeptide of a B cell receptor. Transcription, splicing, and translation result in a light chain that combines with a polypeptide produced from an independently rearranged heavy chain gene to form a functional receptor. Mature B cells and T cells are exception to the generalization that all body cells have the exact same DNA. Well, yeah, it's like another exception to the rule because we've got many exceptions, but yeah, they're yet another exception to the rule that when measured, like when cells, human cells divide, they're identical. No, they're not. Like they those specifically those need to be as different as possible, yet still recognized by the human body. And yeah, you can probably guess that that's probably also why like immunodeficiency happens a lot because that's I guess that's schizophrenia for you. You make multiple personality to hide and then forget about yourself. Start beating yourself up. I mean, I don't know. Like, have you seen Fight Club? Anyways, uh, yeah. For the the therapeutic use of a retroviral vector, assuming you understand the consequences of overstimulating uh, growth and pressing other factors, we have quite we have developed over the years quite efficient viral treatment like gene therapy treatment, but I believe they might be quite expensive or
Probably
exclusive
in
or something.
approval, well, probably in approval limbo, let's say, so you can
Yeah,
sell
approval.
for
That's yeah. That's
have
a
a
good
bunch
way
of
to put it.
have
They're
a
on
bunch
they're
of
on
old
approval
people.
limbo. That's so yeah. So yeah, and insert RNA version of normal allele into retrovirus. You let the we're gonna cover that in the part two of this course. Because yes, there is a part two of this, and it's gonna involve more of like recognizing a phenotype, spotting the genotype with microRNAs, hairpins, and so on. Like I'm gonna spit some fancy methods to target and isolate genes and so on. But yeah, like hopefully you guys are taken well along. If not, mm, I'm always there for some question. I'll answer as many as I can, as best as I can. And if I know that there are like parts covered in previous courses, I'll also refer to it. Not as a get lost kind of thing, but more of a you should probably take a look at those videos. I covered just what you asked. Yeah, stuff like that. Any question on that one before the last time? Good. So we're there. This is the like operon. Um, so you have here the let's say the ABC building block of a plasmid. Uh, you have the like you have um you have the enzymes or the RNA that codes for the enzymes that can downgrade lactose. The whole operon is something I'm still yet to study a bit further in, but yeah, um, the allolactose inducer and active repressor. It's like yeah, if the lactose is present, the repressor will be inactive and the operon will switch on, which
will enable the reading frame to be read and protein synthesis to occur. That's what happens. So on. So you have allolactose and isomer of lactose de- depresses the operon by inactivating the repressor, and in this way, the enzyme for lactose utilization are induced. Basically, what I just said, but I just read the paragraph under it. Uh, and it's like not really important to memorize completely because I'm trying to facilitate this job for y'all. So don't worry, we're getting there, folks. And I done did it. Um, that's it.
I did
That's it,
not
pretty much.
know that lactose was protein to digest lactose was not like done all the time or something. I did not know that lactose triggered the protein to digest lactose.
Yeah, no, it's we have other mechanism that I also will cover, and but that's the most simplistic way of the lac operon is also very uh, widely used. Um,
We
but
up
yeah,
and down
next
regulate.
we're gonna because then there is other sort of operons. There is the tryptophan operon, which is well the TRP, which is another thing. Yeah, well, I'll cover all those fancy ones next time, and I hope it was already fancy enough for today. I hope I did it enlighten some of you, or like make I don't know, like if I could spark some sort of motivation, or at least admiration for part of this field. I'm happy because not only have I just trained my pronunciation and like presentation skills, I am also like serving as a I don't know, like
A
just
teacher.
to be able to, yeah. Oh yeah, well, yeah, a coach, a mentor, but more of a pioneer. That's how I fancy myself doing science. Is like sparking the interest.
Well, I like it,
Cool.
but I gotta.
Yeah, me too.
Well, thanks very much. Very interesting, but like well above my league. But was good listening to you.
Oh, cool. Well, any question on anything you feel is like, yeah, you want to know more about this, just poke me up, like hit me up. I'm always open for any kind of question. It like just like um if it's like super basic questions, please private message me them because I don't. Of course, that I'm quite protective towards the channel like that.
I see.
It's okay to
So
give,
no spam.
yeah. But like any private message with any level um, like question, like I don't even care if it's like primary school level questions. I'll answer them.
Oh, thanks, man.
No problem.
So may I ask why you're actually doing these courses and when is the next one?
The next one is in 1 week, supposedly. Maybe weird shit will happen because apparently it's the season. But no, it's like normally I do this every week. Every week I do like a part of the crash course, which will end with the crash course number 6. When we end with crash course 6, it that's when the actual plasmid club will begin, and we'll have actual. Um, I'd say I'd say professional plasmid creators like they. JPL and his crew is like they've been doing that for years now. They've been I mean, we've got talented people that's gonna present like um, quality placement creation content per se. So
Oh
yeah,
man,
um, I'm so looking
that's
forward to that. The idea of Gabriel was that before starting that, we should make a crash course and record them in videos and so on. So we would like if you know we would try to bring as many people as we can to the same state of comprehension, pretty much say. And that's what I'm doing. Like I'm speeding it up through the curriculum and yeah, my door is always open. Even if I'm sleeping, you can always ask me a question. I'll answer it at some point when I wake up. I
I think that's fine. We're in the same time zone, so that's good.
I can actually vouch for that. I called him at 5 a.m. and
Perfect. Right on time, Sasha.